Afternoon. My name is Kanaha, Assistant Professor, Department of Pharmaceutical Analysis and Chemistry. Today my topic is enzyme. Okay. What are the factors affecting the enzyme activity? First one is effect of enzyme concentration and substrate concentration and effect of temperature and effect of pH and effect of time and effect of radiation and light and effect of enzyme inhibitors. Okay, these are the concentration of enzyme. As a concentration of enzyme increases, automatically the ratio of reaction also increases. Okay, this means enzymatic activity is directly proportional to concentration of enzyme in the system. These are the rate of the reaction. The more enzyme molecules can react with more substrate molecule. So the reaction rate it to be increases. Okay. These are the enzyme concentration. So in enzyme concentration is increases, the rate of reaction also increases. Okay. And this concentration of substrate. If the concentration of enzyme is kept constant and substrate concentration is increases, the rate of reaction also increases. So initially, the rate of directly proportional to substrate concentration, but the substrate concentration is further increased, the remains are unchanged. So we are going to changes of certain changes only. We are also rising the increasing the reaction of substrate. The remains unchanged. As the concentration of substrate increases, the enzyme becomes saturated with substrate. So too much concentration of substrate, the enzyme completely saturated with substrate. Okay, this point of saturation is also rate. Okay, then the effect of temperature. The rate of enzymes reaction increases with increase the temperature up maximum and decline, and bell-shaped curves usually observed. As the 10 about 10 degrees Celsius rise the temperature, it will increase the activity of most enzymes from 50 to 100 percentage. When the enzymes are exposed to light higher temperature, denaturation occur and enzymes lose their activity. Okay, so these are the graph showing the effect of temperature on the enzyme activity. So effect of pH, the increase in hydrogen ion concentration in influence enzyme activity. The bell-shaped curve is a bell-shaped curve uptime. The enzymes are affected by the changes in pH. And the most favorable pH value is 0.1, almost 4 to 9. So in these 4 to 9s are slightly changes, is completely changes to the enzyme activity. So below the optimum pH is much lower and extreme pH enzyme become inactive. Okay. So power pH range is 4 to 9 and effect of time. The under ideal optimum conditions when pH and temperature optimum, the time required at enzyme reaction is less. The effect of light and radiation and the enzymes get denatured and deactivated exposure UV and ultraviolet alpha, beta, gamma rays, etc. High energy radiation from peroxide with oxidase enzyme to make them inactive. So this also factor affecting the enzyme activity. And example, the salivary amylase is deactivated by the UV rays. Okay, and effect of enzyme inhibitors. The presence of enzyme inhibitors reduce enzyme action. Okay. What are the enzyme inhibitors? The enzyme inhibitors are defined as a subset that bind to the enzyme and decrease the catalytic activity that enzyme of the enzyme. This is called as enzyme inhibitors. So organic in basically organic and inorganic in natures, and they are classified with three types. One is reversible inhibitor and competitive inhibitor and non-competitive inhibitor. So first one is reversible and irreversible and allosteric inhibitors. Okay. So these are reversible inhibition. This inhibition bind non-covalently, means big bond with an enzyme's inhibition can reverse if inhibitor is removed. Okay, this two type. This first one is competitive and non-competitive. What means in competitive mean the inhibitors competes with substrate and bind at active site of enzyme. This depend upon concentration of substrate inhibitor. They have similar structure to that of the substrate. This is the competitive inhibition. The enzymes bind with substrate and inhibitor. The enzyme inhibitor form to enzyme inhibitor complex as inhibition bind to the active site. So no reaction occur. So these are non-competitive inhibition. So enzyme react with substrate and inhibitor. The enzyme inhibitor complex formed. The inhibitors bind to the allosteric site. So no reaction the site. Okay, is irreversible inhibition. These inhibitors bind to covalently tightly with enzyme is inactive them. The deactivation is reversible. These inhibitors are toxic and poisonous. Example, penicillin antibodies act reversible inhibitor of serine containing enzyme, black bacterial cell wall synthesis. And these are reversible inhibitors and substrate. And these are competitive enzyme inhibitor and non-competitive enzyme inhibitor. So the competitive means the structure of molecule is similar that substrate. Non-competitive means structure different substrate. Okay, completely different. They better get attached to active site of enzyme. They attach with active site. So form complex pointer than the active site. And it complete with substrate molecule for the free enzyme. It is not a complete with the substrate. This does not alter structure of enzyme. This it does not alter the enzyme or the structure. Then the effect of competitive inhibitor usually temporary is the permanent. Okay. So it's finally is allosteric inhibition by the addition of specific substance, that is activators or inhibitors, the rate of reaction also increase or otherwise decreased. So enzymes which inhibit site of called allosteric site. The allosteric site, the binding with non-substrate molecule, enhance interactivity of the. Thank you.